All right, welcome back, everybody. Today is a really important day because we're going to talk about cell transport. Now, out of any singular topic or thing we've talked about or will talk about in our second unit about cells, cell transport is probably the singular most important one. Not that that other stuff is unimportant, or sorry, not that that other stuff is unimportant, or that we don't need to know any of the other things. But if you were going to sit down for the AP exam and you were going to look at all the different questions that might come from Unit 2, you are most likely to see the highest proportion about this topic, right? Because we're going to talk about how substances get into and out of cells. And substances getting into and out of cells is absolutely crucial in then maintaining homeostasis. So we really, really, really need to know this information forward and backward. So, for example, if you're starting to review the things you've learned so far, this is where I would start. Now, obviously, things that we've talked about previously, specifically in our last lesson about the plasma membrane structure and function, will be directly linked to your ability to understand this section. So, if we want to know about how substances get into and out of cells, we have to know at least a little superficial information about physics or the second law of thermodynamics. All right. What that means is that the universe in general tends towards disorder or entropy, which just means that it tends to move in a direction where the possible state of an individual particle grows over time. What that means is that substances will tend to diffuse, or that means a substance will tend to move from a place where it is more highly concentrated to where it is less highly concentrated. So think about uh, in your classroom. So I remember being in middle school like it was yesterday. And when I was in middle school, this was at the beginning of the 2000s, and what was it, 2002? I was in sixth grade. And Axe body spray was a huge thing with boys, like and I remember we all had our favorite scents, um, and we would wear like literally a ton of it. Like you spray half a can on yourself at the beginning of the day, and somehow we thought that was cool. That was the greatest thing ever. It just smelled amazing, whatever. But you'd go into class and you'd have so much Axe body spray just covering yourself that it would just stink up the whole room because now as I'm in my 30s, I'm like that stuff stank. That no ways about it. I wish there were girls that would have had the guts to just tell us like, dude, this smells. Like just knock it off. But anyway, diffusion's like that where if you spray perfume or Axe body spray into a room, it will move from where you sprayed it to eventually fill the whole room with that scent. It's moving from where it's highly concentrated to all the corners of the classroom where it's less highly concentrated. This is how substances that are able to cross the plasma membrane freely. So I'll give you a second. Let's think about from our last lesson. What are those substances that can freely move across the plasma membrane? If you said small, non-polar, uncharged substances, you're correct. Now, those things can move into and out of the cell membrane by simple diffusion, meaning they're going to move into the cell if they're highly concentrated out of the cell and less highly concentrated inside of the cell, or outside of the cell if the case is reversed. I'm talking oxygen. I'm talking carbon dioxide, those type of things. We call simple diffusion, when things move via diffusion into or out of the cell, a form of passive transport. Because this happens automatically. No energy is needed. They're going to move from one side to the other just via passive diffusion, simple diffusion. So this purple substance will eventually diffuse to fill this whole box. Or these green dots will diffuse through the membrane if they're permeable to the membrane towards the left side through simple diffusion. Uh, the cell membrane is the boundary between the inside and the outside of the cell, and it can allow certain substances into the cell and certain substances like waste out of the cell. So a cell needs to make sure that it can take in the substances it needs, like macromolecules and water and oxygen, and get rid of ammonia and salt and carbon dioxide and these other things outside of the cell. Now, some of those things will move in and out because of the concentration gradient through simple diffusion. Others will be a facilitated diffusion, so things that are larger or charged or nonpolar that need to move in and out of the cell that can't just go through the plasma membrane on their own because the membrane isn't permeable to the substances, will diffuse through a protein channel or gate. And this is called facilitated diffusion. And again, there is no energy needed for this because it just happens via diffusion from high to low. But they need protein channels or doorways to provide a conduit through the membrane for those substances that normally couldn't, they're large or polar, to move into or out of the cell. Now, there's also another transport mechanism called active transport. Now, if you're active, you have energy. So this type of transport requires energy. And it requires energy because this is how cells move substances against their concentration gradient or in the opposite direction of diffusion, from low to high concentration. And they do that through protein pumps. These protein pumps physically carry or change shape to move or force substances into inside or outside of the membrane where they're already concentrated. And because they're trying to counteract uh, diffusion or facilitated diffusion from happening, they require ATP energy. So this comes at a cost, right? That comes at a cost. Here's an example of a protein changing shapes or having a conformational change to allow a substance to bind to it and then push it in the direction it wants to go. There's many different types of active transporters. There are symporters that might move two things in the same direction. So here, amino acids and sodium are being pushed in the same direction. Or they might have antiporters, in which case, in this picture on the left, amino acids and sodium are being actively transported in opposite directions. So one is at a high concentration out of the cell, and the other is a high concentration in the cell. So in general, there's two forms of transport, passive and active. Passive consists of simple diffusion and facilitated diffusion. The main difference between those two is whether a protein channel is needed or not. And active transport, which requires energy that moves substances from low to high concentrations instead of from high to low concentrations. So here's a picture that summarizes those three mechanisms and how we group them from active to passive transport. But what about large molecules or large amounts of molecules at a time? Moving large molecules or large quantities of molecules into and out of the cells through vesicles and vacuoles through endocytosis and exocytosis. So in endocytosis, substances that are going to be taking endo into the cell move towards the cell membrane, and the cell membrane starts to form a bubble around that substance. And that bubble grows bigger and tighter until it pinches off and eventually floats freely inside of the cell in a vesicle. So phagocytosis or cell eating, and penocytosis, cell drinking can occur. And there's also a process called receptor-mediated endocytosis, where the substance is only moved into the cell in a vesicle when it attaches to a protein receptor on the surface. And the binding of the substance to the receptor on the surface causes the formation of this vesicle, like you see in this third picture. Exocytosis is the literal reverse of that. So you have something in, I'm going back to the past slide. You have a vesicle full of maybe like waste that you want to spill out of the cell, and that vesicle fuses with the membrane and then spills its contents out into the extracellular environment. So endocytosis and exocytosis are main active transport mechanisms for moving large quantities of substances into and out of the cell. Now, lastly, we need to talk about the special case of water. Now, movement of water into and out of the cell is so important and so special, it gets its own name. And we call that osmosis. And osmosis is just the diffusion of water. So everything is happening the same as diffusion. Water is moving from a high concentration to a low concentration. No energy is required. We just call it osmosis because it's so important for, towards a cell. So the direction that osmosis will occur in is determined by um, the amount of water. So let's look at this picture below. These yellow dots are dissolved substances or solute, and the water is the solvent. So review chemistry time, right? A solute is the substance that's dissolved, and the solvent is the thing that the solute is dissolved in. So in cells, the main solvent is water, and the solute might be sodium or chloride ions or something dissolved in that water. Now, if I have, for example, a system set up where there's a membrane, maybe this is out on the left is outside and maybe on the right is inside of the cell, and this line here in this picture is the cell membrane. The side of the membrane that has more dissolved solute, so the right side of this membrane has more dissolved solute than the left. There's more dissolved stuff than the left. Notice that if there's more dissolved solute taking up space, then there has to be less water, right? Because it's the same amount of space. So if there's more solute dissolved, there's less water. And opposite, on the outside of the cell, on the left side of this picture, there's less dissolved solute, less yellow dots, so there's more water. If there is, like the left side, less dissolved solute, so there's more water, we call that hypotonic. So we would say this left side is hypotonic to the right side. The left side has less dissolved solute particles, so more water. We would say it's hypotonic to this right side. Now, this right side has more dissolved solute molecules, more yellow dots than the left side. So there's more solute and less water. We call it hypertonic. So water will always move from a hypotonic, so from the hypotonic side, where there's less solute and more water, to the hypertonic side, where there's more solute and less water. It's still moving from high to low. Hypotonic, more water. Hypertonic, less water. Still low to high. Just simple diffusion, but from the hypotonic side to the hypertonic side. Now, if there was the same amount of solute and therefore the same amount of water on both sides, we would call the two sides isotonic, and there would be no net movement of water in either direction. So we need to be okay with using these terms, hypertonic, hypotonic, and isotonic, to compare two different solutions. They are only useful from comparison. So, for example, this hypertonic side is only hypertonic compared to the left side. I might put a third box here, even farther to the right, that might be even more hypertonic than that what's labeled as the hypertonic side. So just because this side is labeled hypertonic compared to the left side, it might be hypotonic compared to a third solution. So these terms are only useful for comparison relative to one another. So managing the water balance or how much water is moving into and out of a cell is super important for all life. Cell survival depends on balancing water uptake and loss. So, for example, if I would put an animal cell and a plant cell into a hypotonic solution, water would want to rush into these cells. In an animal cell where there's just a flexible plasma membrane binding it, water could rush into the cell so fast that the cell explodes at lysis. In a plant cell, there's got the rigid cell wall on the outside as well. So the cell's not going to burst or lyse. It's just going to become really stiff and turgid, just like a plant, like a potted plant after you water it. Okay. This is one of the main reasons that if you go to the hospital and they give you an IV, it's not of distilled water. It's of normal saline, which is of 0.9% sodium chloride solution. If you would get an IV of just pure water, all that water would rush into from out of your blood into your body cells and they'd lyse all over the place. So they put in normal saline solution so that it is the same osmolarity or the same concentration as your normal body tissues. So it's an isotonic solution. On the other hand, here's a plant cell and an animal cell in an isotonic solution. The animal cell will, of course, just stay its normal size because as water goes in, an equal amount goes out. And a plant cell is will just be a flaccid or a limp cell. This is kind of what you would see if you didn't water your plant often enough. Now, on the opposite side, if we put animal and plant cells into a hypertonic solution, water is going to want to leave the cell. In an animal cell, it will shrivel up. And in a plant cell, since we've got that cell wall, it can't really shrivel because it got a rigid cell wall there. It will plasmolyze, which means the plasma membrane will start to shrivel and actually pull itself away from the cell wall. Now, hypertonic solutions are actually super important sometimes in medicine. I'm actually a registered paramedic. I did that because originally when I was going to go into medicine, I was planning on being a pediatric intensivist, and I thought that getting some experience with emergency medicine would be important. But I still work as a volunteer as a paramedic in Deerfield, in Oregon, a little bit, not as much as I used to. But I used to volunteer for the town of Madison Fire Department, which is actually going out. It's getting taken over by the city of Madison. And one time that we had a section of the Beltline that was part of our jurisdiction, what we had to respond to calls with. And there was, yeah, I can never forget it. It was in the winter. It was probably like February. Let's see, probably like 6 years ago, like 2018, 2017, 2018, the winter there. And there was a really bad accident. And we got called out there and it was like 2, 3 in the morning on a Saturday morning, like 2, 3 a.m. And there was a patient who had been in this car accident and suffered this really severe head trauma. Skull was fractured, things like that. And the issue with that is that if you suffer severe head trauma, the brain itself will start to swell. Okay. And it will swell, and so it'll get bigger and bigger and bigger. And the danger with that is your skull is kind of a closed container that's filled with fluid called cerebrospinal fluid. And fluids don't compress very easily. So as your brain swells, because that fluid doesn't compress easily, the pressure inside your cranium builds and builds and builds and builds. And one of the really dangerous issues with that is if your brain starts to swell after a traumatic episode and that pressure increases, 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 it could increase to the point where the base of your brain, like where the brainstem is, gets pushed out of the this hole in the bottom of your skull, kind of by where the spinal cord attaches to, called the foramen magnum. And it's called herniating your brainstem. And that could cause brain death, like within a short amount of time if that herniation occurs. So one of the pre-hospital treatments for that is to give us a patient an IV with a substance called mannitol, which is a hypertonic crystalloid substance. And that hypertonic solution pulls water out of the cells into the vasculature of uh, of the patient. And so it pulls water out of the cells and thereby decreasing the pressure in their cranium and allowing us to be able to have more time to transport them to the ER and the ED so that they can be treated without risking herniating their brainstem and that being deadly. So that's a really good tie-in to the hypertonic solutions. So here's just another example. These next slides just show you a little bit more about what would happen if we put plant and animal cells in hypotonic solutions here, um, and so on and so forth. Um, Organisms that live in water, like paramecia, constantly have to battle this, and they have, like we mentioned in our last or our second lesson about eukaryotic organelles, they have vacuoles that sole job is to pump out water because they're never going to become, since they live in water, they're never going to be able to not have water constantly moving into their cell. And so basically, their only way to survive is have this vacuole that continually pumps water out into their environment. Just like if you want to keep your house cool in the hot summer, you have an air conditioner, right, that constantly pumps in cool air and pumps out hot air. Here's another slide where it looks more specifically over hypertonic solutions. I'm not mentioning as much because we kind of really talked about it there when I went on my stories, on my tangent a little bit earlier. So you can pause the video and look over this if you'd like and with isotonic solutions, right? And I'm not minimizing the importance of it. I just prematurely started, talking about these and going on and said everything that I needed to say. And so here's a good slides that reinforce those things that you can pause and you can look at them if you're watching this video. Okay, kind of lastly, aquaporins were a really important discovery in the late 90s and early 2000s that allow water to be moved rapidly into and out of the cells. So there were evidence that there were water channels or proteins that allowed water to flow across the membrane more readily than they normally would because water is a small but polar molecule. And these two gentlemen actually won the Nobel Prize in Physiology or Medicine for the discovery of aquaporins. So let's just check. Do you understand osmosis? We've got this cell that it has a zero point 0.3 molar sucrose and zero point 0.2 molar glucose on the inside. And so it has a total solute concentration of zero point 0.5 molar. And we have an environment that has a total of zero point 0.3 molar. And we'll say the cell is completely impermeable to carbohydrates. So this membrane cannot allow sucrose or glucose or fructose to go through. So hopefully we know that the cell, compared to its environment, the solution is hypertonic. And the beaker compared to the cell is hypotonic, so water would flow in to the cell from the environment, the hypotonic solution to the hypertonic solution, right? Right. Thank you very much for your time today, and this is what we'll be working on in class.